Hello, everyone. My name is Somayeh. I'm on behalf of my team, Abbas and Mina, which Mina is a supervisor and Abbas is a PhD student. I'm working closely with Abbas for developing a techniques for microplastic detection. So, is this working? No. Oh, it's not. But just to start, where the story of microplastic detection in our lab started, it was not our intention to work on microplastic. We had a background in the biosensors and kind of the gas sensors, but not microplastic. The story started when an industry partner approached us. They were developing and fabricating coffee machine. They had such a concern that if there is any leach of the microplastic through the brewing of the coffee to their drinking coffee or not, which was a very valid point because when we investigated the design of the device, we figured out that the internal part of the device is made of the plastic, the most of the hot water combination with the coffee grains happening over there. So we did kind of investigation through the regular techniques, as we know, for example, SEM imaging, and this kind of the techniques, when we took the sample from the coffee brewed through the device, we figured out that there are kind of the pieces of the plastic, which is through the Raman spectroscopy was a validation. They're coming through the scratch of the internal part of the coffee machine. So we did so much imaging with the help of the co-op student, PhD student, and through collecting the SEM images with the validation across validation with the Raman, we started calculating the surface area of this plastic, which we know that the type of the plastic, which is showed, this is the internal part of the coffee machine, and these are the scratches through the brewing happens. And we find that approximately per cup of the coffee per day, a person can get 33 milligram of the microplastic through the drinking of the coffee. So that was a great concern over there. But it was an initiation for us to think about that microplastic is a real concern. We should take more action about that. We shared this kind of the things with the industry partner. They were just trying to redesign their coffee machine by adding different materials for the core part, or just coating with some kind of the ceramic materials which are biocompatible and they are not made of the plastic. But that was the where, and we started continuing our research on microplastics. So it was the time to use our knowledge in the sensor development for detecting microplastics. For that purpose, we know that there are so much concern about the water contamination coming from the leach of the plastic through different sources into our drinking water, different source of the water like ocean water, river water. So we use our technology, whatever we know in the sensing world, based on the, for example, on the kind of the corner that shows the combination of the Raman with microscopy techniques. That was the kind of the gold standard techniques that is very good to be used, but they are not a kind of the handheld miniaturized systems. Most of the industry partners, we worked with them and we're interested in microplastic detection. They wanted a small size miniaturized techniques for detecting microplastic. So we were thinking to develop another techniques. We just tried different techniques like triboelectric, electrochemical, and also peptide, a selective peptide interaction with the microplastic. For today, I just focus on the peptide things. And as you know, a while ago, as I showed on the right corner, people were using molecularly imprinted polymers for selective separation of the protein, specifically peptides. That was used a while ago. And after that, we were thinking why we, group of the researchers were thinking why we do not do it reverse action. We take the peptide as a source for bonding selectively to plastics for the plastic detection. So we took this opportunity for developing techniques based on the selective interaction between peptide and different type of the microplastic. The point about the different type of the microplastic is that peptide is made of the amino acids, and you can design and engineer this sequence of the amino acid that they can make different interaction with different plastic types. So you can engineer your peptide to be selectively only bind to the specific type of the plastic. And there are different type of, back to the chemistry, this kind of interaction can be electrostatic and a kind of non-covalent kind of interaction. And that's the way we develop our sensor. So for the first part, we asked a team that they are great in synthesizing peptide. They design a sequence of the peptide that have the affinity to bind to the polystyrene, PS, and PP. So for that, first of we ask them to just tag them with the fluorescence. That can give us a kind of confidence that this kind of the interaction happens through the, when we mix them in a water sources, and if there is a microplastic, this kind of the peptide has the affinity to bind to the any PS or PP probable potential presence of this kind of the plastic type. So as the images of the fluorescence microscopy shows that these kind of the particles, a spherical shape, are the intentionally added polystyrene particles we added to water sources, and we mix them with the peptide sources tagged with the fluorescence. As you see that the syringe, the kind of the peptides are just bonded around this microplastic, and because of the fluorescence tag, we can distinguish them on the fluorescence microscopy. We did kind of the test to find out what is the optimal incubation time in terms of the temperature needed for optimized interaction between these two, and also what would be the best time of the incubation. So this kind of the things optimized, we figured out that even at room temperature, just 5 minutes is enough to mix these two together and find out the best optimum interaction, and we see them under the microscope. So later we move to, these are the SEM images also for the validation to show that these are the PS particles, spherical shape microbeads before interaction with the peptide, and after being interacted with peptide, you see the coded part of with the proteins of the peptide, and also different images based on the size of these PS particles. So later we moved on because this kind of the techniques should be practical in the real world. If you use the seawater, that seawater has so much ionic and so much ions, and also the other things and stuff in it. So how selective these things can bond it? We use the source of the seawater. There are plastic that in different shapes and different kind of the sizes were added intentional to the seawater. We mix them at the same incubation condition to the peptide things, and we saw in the fluorescence microscopy that there is a selective bonding to the plastic, and we couldn't see the rest of the things under the fluorescence microscopy. Also, after drying, these are the salts, dry salts of the salty water of the sea. And you see still they are stable. So the environment of the seawater doesn't impact the interaction between the peptide and also microplastic. It doesn't matter what would be the shape, what would be the size, the morphology. This interaction happens very easily through the mixing the peptide with the water sources. And I'm sorry that I'm going mistake. This is the first publication. We go through that, but it was not unintentional because the fluorescence looks good in here, but it's a very bulky machine, and everyone looking for something to be a small miniaturized, and they can be deployable. They can use it on site. Specifically, for example, we had an industry partner wanted to develop something for camping person. If you go to the camp, you just wanted to try your drinking water is clean or not. It's just a yes or no. So that's the things that the fluorescence was good as a first initiation validation of the developed technique, but was not enough for our purpose. We moved to the electrochemical measurements, but electrochemical measurement was good. We just coated the steam-printed electrode with peptide and used different source of the water. It was good as long as your water is clean, means drinking water or very pure kind of the water. But when it comes to the seawater, because of the high ionic environment, and the whole fact of the electrochemical is based on the electron charge. So high conductivity of the seawater doesn't help for measuring through the electrochemical. It was not successful for the real world samples. So we get to another techniques, UV visible spectroscopy. What we did, we used the techniques of the peptide, but in here we asked our collaborator to add AU tags instead of the fluorescence tag. The point of that is the AU, when the peptide bond to the microplastic, these nanoparticles of the AU can aggregate, and it causes the color of the water changes. And we can visually see through the change of the color if there is any microplastic or not. So it's a yes or no techniques. But in advanced level, we can use the spectroscopy measurement to make it more quantifiable. So this is the quantifiable version. This one shows the absorption spectra related to each of the elements we plan to mix together: peptide, polystyrene, and also gold nanoparticles. When we take the spectrum of the gold as the blue ones, and when we mix them just only gold and microplastic, no interaction is happening there, and the gold wavelength is at the exact place that it should be. So it means that there is no random affinity between the gold and microplastic. But when we add the peptides, this kind of the bondings, because the gold has a cysteine, has an affinity to bind to the one head of the peptide and the other end of the peptide, bond to the microplastic, and all this combination and aggregation shift the wavelength because it changed the color to the reddish color, which goes to the higher wavelength of the absorption. So that's the kind of the way we could detect it through the spectroscopy-based technique, both visually as a yes or no, or quantifiably through this spectrum measurement. The whole technique's mechanism is based on that the gold nanoparticles, when they are individually, they have their own specific absorption wavelength. But when they are bonded to peptide and aggregate with the gold metal, plastic types, because you're aggregated on the surface of the microplastic, first of all, their refractive index media will be changed. Second of all, because of the coupling between the frequency of the resonance or the resonance plasmon, we call it, this both combination cause the wavelength of the absorption shift to the higher wavelength. That's the way we develop this technique for the detection of the microplastic. And further, we analyze based on the size. As the size increases, the shift gets to the higher wavelength, and also we analyze based on the different concentration. And by increasing the concentration, it goes to the higher. So it means that higher particles have higher surface area, more peptide bonding happens. So we can expect this higher shift going to the higher wavelength, and also higher concentration is also again providing higher surface area, more bonding happens, and we can expect higher shift to the wavelength. Also, we tried how selective these techniques could be, because whole the peptide things, as I told you, was designed specifically for PS and PP. We exposed it to the PET and nylon as another common type of the plastic in the source of the water. We saw that, as shown here, they just show the shift to the polystyrene and PP, not PET and nylon. So they selectively are designed to bind to those type of the plastic. And all the tests I so far explained, they were done in the clean water. Now we just wanted to try to see all these things that we explained can be work in the real seawater or not. So we repeated all the tests in the seawater, as shows the graph, the blue line and as a dear water, and the orange one as a seawater, just kind of follow each other in a linear way. So there's a kind of the similarity between them. The only difference is that because of ionic environment of the sea impacts the absorption and these things, but the whole story is kind of the very similar between these two. So it shows the interaction is stable and it's working in the real case scenario. Also, we did a kind of data analysis. PCA clustering shows that on the top, based on the color code, they are the size of the particles in different concentration, and as we go forward, the size increases, and also we have different concentration for each particle size. We see the trend increasing in the trend, but as you see, there's a kind of overlap. For example, smaller particles in higher concentration are kind of similar with the bigger particles in the lower concentration, because the surface area kind of very similar to each other. So what impacts in here, how much surface area is available for the peptide to bind to the microplastic. This is a kind of the challenge for our developed techniques that we should figure out, because it's not for now accurately can distinguish between the particle size and the concentration range. We are working on it, but so far, data analysis can help us. Even a small difference between the shift of the particles can be distinguished and be analyzed further for the detection. To summarize what we said, this kind of the technology from the story of the coffee machine get us to collaborate with international universities like University of Iceland, and through them we get to know a famous swimmer called Ross LG. Ross LG, this last April, started swimming around the Iceland, and he had so much message to the world. One of the message was the concern about the microplastic. On behalf of University of Victoria and Mina team, he's sending this message to the world. And also he did us a favor. He just from different spots tried to take the samples. Those samples are just transferred to our lab. And we are planning to test our developed techniques based on the real samples he has provided us. And thank you so much.